Hello everyone, my name is Bhavita. I would like to explain about CRISPR. Genes determine our traits like eye color and freckles. Some genes make people more likely to get certain diseases like high cholesterol or some cancers, whether it's cancer, HIV or antibiotic resistance. So much of modern medicine is influenced by people's genes. Using a technology called CRISPR, scientists can edit the DNA of the genes. CRISPR helps scientists to locate a faulty gene, then cut the DNA strand, whether it's located to remove, fix, or replace it. Genome editing is a form of genetic engineering in which DNA sequences are inserted, deleted, or replaced in the genome of living organisms using molecular scissors like nucleases. The four main families of nucleases are meganucleases, zinc finger nucleases, transcription activator like effector nucleases, and CRISPR. CRISPR cluster regularly interface short palindromic repeats is a prokaryotic immune system that confers resistance to foreign genetic elements such as those present within plasmids and phages that provides a form of acquired immunity. It forms the basis of a genome editing technology known as CRISPR-Cas9 that allows permanent modifications of genes within organisms. CRISPR-Cas system consists of two components: the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited to use CRISPR-Cas9. The CRISPR story began with the discovery of a peculiar short repeat in the E. coli genome by Ishino and co-workers. Subsequently, similar repeats were noted in bacteria in 1987, and in 2002, Mojica and Janssen coined the CRISPR acronym and characterized CRISPR locus. First experimental CRISPR adaptive immunity was done in 2007. In 2012, idea of using CRISPR-Cas9 as a genome engineering tool by Jennifer Doudna and Emmanuel has been done. In 2013, demonstration of Cas9 genome in eukaryotic cells. In 2015, the CRISPR-Cas system was selected by Science as 2015 Breakthrough of the Year. The CRISPR system is a highly adaptive and heritable resistance mechanism that incorporates sequences derived from the foreign element into a small RNA-based repertoire. The small RNAs program an enzymatic complex to recognize and destroy the invader. Different CRISPR-Cas system in bacterial adaptive immunity. In type 1 contains Cas3 gene which encodes large proteins with separate helices and DNase activity in addition to genes encoding protein that form cascade like complexes with different compositions. In type 2 includes HLH type system in which Cas9, a single very large protein, is sufficient to generate cRNA and leaving the target DNA in addition to ubiquitous Cas1 and Cas2. Type 3 contain polymerase and ramp modules in which at least some of the ramps seem to be involved in the processing of the spacer repeat transcripts. A CRISPR locus is defined as an array of short direct repeats interspersed with spacer sequences within a given locus. The repeats are practically identical in length and sequence. The spacers are also uniform in length but have highly variable sequence content. Among different species, repeats vary from 21 to 47 base pairs, being 32 base pairs on average. Spacers are of a similar size. Related species can have similar repeat sequences, but the overall bacterial and archaeal sequence diversity of both spacers and repeats is great despite their sequence diversity. A majority of CRISPR repeats has the potential to form apparent structures. Here is a picture of a CRISPR locus. Key components of CRISPR. CRISPR RNA contains the guide RNA that locates the correct section of host DNA along with the region that binds to tracer RNA. Tracer RNA, transactivating RNA, binds to CRISPR RNA and forms an activate complex. gRNA, guide RNA, a combined RNA consisting of activating RNA and at least one CRISPR RNA. Cas9, they are RNA-guided DNA endonucleases associated with CRISPR to interrogate foreign DNA. Mechanism and action of CRISPR. Editing a gene using the CRISPR-Cas9 technique. Scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular sequence in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9. Then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA at the point. Cas9 has amazing ability to recognize a particular DNA sequence in a cell that is faulty. Then the guide RNA homes in on the target DNA sequence and Cas9 cuts it out. Once the job is complete, the guide RNA and Cas9 leaves the place. Now another piece of DNA is swapped into the place of the old DNA and enzymes replace the cut. What makes CRISPR system the ideal genome? CRISPR is a unique technology that enables researchers to edit parts of the genome by removing, adding, or altering sections of the DNA. It is currently the simplest, most versatile method of gene manipulation. Why is CRISPR-Cas9 better? It works speed, accuracy, and low at price and simplicity. Applications of CRISPR in modern medicine. It helps to understand the role that specific mutations in specific genes influence a particular trait of an organism. It helps to recreate known stable mutations in cell lines that can serve as models of a particular disease. It also helps to create stable mutations in whole organisms, in plants or animals, and create strains that can be used in research or commerce. to create gene therapies in order to treat or diagnose congenital diseases, infections, or cancers, especially new outbreaks like Zika virus, to create gene drives that can modify populations of organisms in a specific way. What can we do with CRISPR-Cas9? Deleting a gene, adding a new gene, activating dead genes, controlling gene activity level. Deleting a gene. Undesirable genes can be deleted from the genome, allowing researchers to study their function-specific genes and learn about what happens to the cell when these genes are not in the genome. Adding a new gene. Desirable genes can be added into the genome, allowing researchers to study their functions within the cells. These genes can also add new functions to the cell. Activating dead genes. Genes that are essential for various functions but no longer functions can be reactivated using CRISPR-Cas9 system. Controlling gene activity level. Genes are more active than normal, can control to be produce just the right amount of proteins, which will help maintain the balance within the cell under desired conditions. CRISPR treatment for DMD. DMD, Duchenne muscular dystrophy. New gene editing enzyme CRISPR corrected DNA in human cell. CRISPR copy differ from CRISPR-Cas9, much smaller than Cas9 enzyme, which makes it easier for the delivery to muscle. Skipping a mutation region or precisely repairing a mutation in gene, CRISPR-Cas1 mediated genome editing corrects DMD mutation but also muscle contractility and strength. CRISPR targets point mutation in exons as stop codon and serve as a model of DMD. Efficacy by using a two-vector system of CRISPR rather than single vectors of the guide RNA and Cas9, which will restore the dystrophy positive fibers. Recovery. Cell level decrease infiltration, inflammatory cells and decrease fibrosis and reversal of necrosis. After the mechanism, the final outcome: improved grip strength, force generation, resistance against contractions, decreased blood levels of creatine kinase, dystrophin expression on vascular smooth muscle, important source of oxygenation during activity, and cardiomyocytes restored dystrophin. Rescue of dystrophin expression. In normal muscle, it appears like this. In dystrophic muscle, it appears like this. After the mechanism of CRISPR, the corrected muscle. Conclusion. The CRISPR-Cas9 technique is one of the number of gene editing tools. Many favor the CRISPR-Cas9 technique because its high degree of flexibility and accuracy in cutting and pasting DNA. One of the reasons for its popularity is that it makes it possible to carry out genetic engineering on an unprecedented scale at a very low cost. How it differs from previous genetic engineering techniques is that it allows for the introduction or removal of more than one gene at a time. This makes it possible to manipulate many different genes in a cell line, plant or animal very quickly, reducing the process from taking a number of years to a matter of weeks. It is also different in that it is not species specific, so can be used on organisms previously resistant to genetic engineering. It helps in human health, new materials, drug development, research applications, in agriculture and in bioenergy. CRISPR-Cas9 system will revolutionize gene therapy and make it possible to treat large number of diseases that would be impossible to treat without this technique. This includes diabetes, cancer, cystic fibrosis, and sickle cell anemia. In new materials, manipulating biological circuits using CRISPR-Cas9 will facilitate the generation of useful synthetic materials that could be useful in various applications such as oral drug delivery and the production of biosensors. In drug development, engineering cells to optimize high-yield generation of drug precursors in bacterial factories could significantly reduce the cost and accessibility of useful therapeutics. In research applications, CRISPR-Cas9 system will allow the creation of new animal and cellular models which will help us learn more about diseases, test new drugs and vaccines on these models. In agriculture, CRISPR gene editing tools can edit crops without harming other genes, which will help confer resistance to infections and harsh environments, improving global food security. In bioenergy, sustainable and cost-effective biofuels are attractive sources for renewable energy, which could be achieved by creating efficient metabolic pathways for ethanol production in algae or corn. And hence, this is the future of genetic engineering, the CRISPR technology.